The overall goal of this procedure is to use RNA interference to investigate the effect of candidate genes on the innate immune response in mouse macrophages. To do this, mouse macrophage cell lines are trypsinized and combined with transfection solution and the siRNAs of interest. The mixture is then transferred to a 96-well nucleocuvette plate. An Amaxa 96-well transfection system is used to transfect siRNA into the macrophages. And after an incubation with cell culture medium, LPS or other activators of innate immunity are applied to the transfected macrophages to stimulate them. Finally, ELISAs are then performed to monitor cytokine production and assess the effect of the siRNA treatment on the innate immune response.
The main advantage of this technique over lipid-mediated transfection is that our assay is robust and can be applied to many genes in high-throughput fashion.
The key to this method is to work with healthy cells, treat them gently, and work efficiently during the transfection process, as we will demonstrate.
This protocol uses the raw 264.7 mouse macrophage cell line, which is cultured as described in the accompanying document. In preparation for the siRNA transfection, plate the cells at 20% confluency and grow 1 or 2 days until the cells are no more than 80% confluent. It is important to remember to use cells that are between passages 3 and 10.